I'm gonna kind of speed through this lecture because, yeah, we have a little less time than expected, but I'm gonna talk about brain imaging techniques. Thanks to the groups that presented today, and sorry we didn't get to the third group, kind of ran out of time. Yeah, okay, brain imaging techniques. So this is kind of our lecture outline that we're gonna go through today. We're gonna start with some history. So this guy is Franz Gall. He is kind of considered the father of functional brain imaging. He postulated that the localized region of the brain has a special function and that the integration of these parts together is what gives rise to complex behaviors. Unfortunately, he was a phrenologist, so he thought that like personality and morality and intelligence were correlated to the shape and size of the skull, which we now know it isn't. But he was like kind of one of the pioneers of brain imaging, so gotta give him the credit. Okay, so we know that organization and networking of the brain is what gives rise to human behavior and function. So like, how is this useful? Just like throw out some ideas, like why do we care about the function of the brain? Like how can we use it? Any ideas? This is a Neurotech decal. It's a big clue. Okay, anyway, whatever. All right, disease treatment, apps, or like human neural enhancement, those are all things that we'll get into. Okay, so a couple challenges with brain imaging. So first of all is the skull barrier. Obviously there's this layer of bone between, the external world and your brain. And so like, it's like a physical barrier that will literally block imaging, visualization, like going into the brain itself. And then also there's unique mapping amongst individuals. Like your broca area might be in a different place where mine is, and like your something cortex might not be the same as mine. So, yeah. So how can we overcome these challenges? Obviously, you can like crack open the skull. Not ideal for the patient. No one's gonna hammer to them, but there are ways to do that. And then also you can like stick electrodes into the brain. Use magnets, you can track with chemistry. These are all things we'll go into in the rest of this lecture. So what's brain imaging? It's basically a means of exploring the overall structure of the brain and the function as well in certain techniques. So it's not only specific brain regions, but also about the connections and activities between them. It allows us to study neural activation at like a population level. So another really important thing in terms of technology is functional neural imaging. It's not only looking at what the brain like literally looks like, but also the mental function associated with activities. So what are some applications of brain imaging? First of all, locating brain damage. Before brain imaging, this was obviously really difficult because brain injuries are really imprecise. And to locate the damage, neurologists often had to like rely on post-mortem examinations, which so like after the patient has died. But obviously those don't necessarily reflect injury at the time of diagnosis, so having like current brain imaging techniques now is really important. Also understanding the brain and its functional activity, like with fMRI, diagnosis in general of medical diseases. One case is the Prader-Willi syndrome, which is it's a genetic disorder that causes weakened muscle, poor feeding, and slow development. And basically a symptom is like non-satiation when eating, which can lead to obesity and diabetes. And through brain imaging, researchers were able to find out that like this disorder has like a neurological basis in the brain. If you can tell from these pictures, the control brain, the frontal lobe, is much smaller than the PWS one, and like seeing that is able to, or like helps scientists like discover this correlation that like there's this increase in neurological activity after eating instead of a decrease like with regular patients. Okay, so we're gonna go into EEG first, which might not be like the first thing you think of when you think of brain imaging, but it's really important for this class because you guys are gonna be diving into EEG data sets and signal analysis and stuff, so we really wanna go into this and make sure you guys have a good understanding. So EEG stands for Electroencephalography, which is why we call it EEG and not that. So EEG measures electrical activity, tracking and recording brainwave patterns. So Basically, neurons are conductors of electrical signals. They're constantly exchanging ions to maintain resting potentials and also to propagate action potentials. So when ions are pushed in a wave, like through the neuron, and they reach the electrodes on the scalp, then they can cause changes in voltages in the electrodes, and EEG is what results from recording those voltages over time. So EEG doesn't actually directly capture like axonal action potentials. They capture the neural back propagation, which is kind of what I described just now. It's something that occurs after action potential of the neuron creates a voltage spike down the axon. And looking at irregularities in EEG data can show signs of brain disorders, which is important for diagnosis. So the hardware of EEG is electrodes, obviously. They're small metal discs attached to the scalp or in the brain. And these analyze the electric signals in the brain and send the data to computers that record the results. There are specific rules and maps that specify where electrodes should go on the scalp. And this picture is called the 10-20 system. So it's basically where distances between adjacent electrodes are either 10% or 20% of the total back to front or left to right distance of the skull. And this method was developed to standardized testing, basically. And it's based on the relationship between the location of an electrode and the cerebral cortex. And also gels are used a lot because electrodes require conducting gels on the skin. It's generally dispensed into the electrode cap or onto the individual electrode, but these aren't that great for long-term recordings because these gels are usually water-based, so they dry out and degrade the quality of recordings. So just some types of brain waves that EEG can pick up. There's 5 of them. They kind of range from being in a peak mental state of a lot of parts of your brain are being used to being in very, very deep sleep. This is something that you can kind of see later on with the data sets you guys are working with the Neurotech headset, which probably won't have time for a demo today, but it's something that you can see from your own brain. Okay, yeah, and then there's invasive and non-invasive methods of EEG as well. So invasive, obviously, the electrodes are implanted on the surface of or deep within your brain. These can be used to monitor seizures, treat epilepsy, or Parkinson's. Like the group before described, deep brain stimulation is electrodes are planted deep into the brain. It stimulates certain regions of the brain, and that provides relief for Parkinson's symptoms like the tremors, etc. This is more accurate than non-invasive techniques because it's directly in your brain, so there's not going to be as much noise. And then non-invasive is electrodes attached to the scalp, so on the outside, above the skull barrier. And this can be used to track irregularities in brain activity. And compared to the invasive one, it's less accurate because of noise. The skull barrier, like you said, blocks signals, so the signal can be a bit fuzzy in the recordings. Okay, applications are largely diagnostic, so usually used in a medical setting. It focuses on the analysis of event-related potentials, which measure brain response that's a direct result of specific sensory, cognitive, or motor events. This can be processing event-related potentials of things like blinks or jaw clenches, or the spectral content of EEG, which is the distribution of power into frequency components making up a signal. So this can be used to diagnose epilepsy, sleep disorders, depth of anesthesia, coma, and brain death. And obviously there's a lot of software involved for analysis, and like I said before, it's something you guys will all be exposed to and working on in the coming lectures. Demo we don't have time for, sorry. Okay, EMG. This is another thing that is not typically thought of in terms of brain imaging, but once again, a data set you guys will be working with. So what is EMG? It's a procedure that assesses the health of muscles and motor neurons that control them. So the electrode basically transmits electrical signals that causes muscles to contract, and then a needle electrode is inserted directly into the muscle that records and translates those electric signals. So the brain has upper motor neurons in the cortex of the brain, and these map to lower motor neurons in the spinal cord, which then have axons that come in contact with individual muscle fibers. So it's got that whole connection from the brain to the muscle. And these axons release neurotransmitters that turn muscle fibers on and off. And yeah, so that's basically what EMG is like exploring. What it's analyzing is that connection from motor neurons to your muscles themselves. And yeah, it's basically listening to our motor planning system, and it's really relevant and important in terms of like, let's get that for now, these kinds of current uses. So like diagnosing muscular dystrophy, which is a muscle disorder, and myasthenia gravis, which is a disease affecting nerve-muscle connection, which kind of makes sense with EMG. It's like looking at the nerve-muscle connection, so it's really good for that. Polio also is a disease affecting motor neurons in the brain and spinal cord, and then carpal tunnel, actually, a syndrome affecting peripheral nerves, can be diagnosed using EMG. Okay, MRI is probably one of the most popular brain imaging techniques in terms of just like what people know about. How many of you guys have had an MRI before? Okay, yeah, a lot of people. Cool. I have, but it wasn't because I had any injury. It was literally because I got paid $200 to be in psych study, and I just got to sit in an MRI for 2 hours. Pretty weird. Okay, I'm gonna skip this video for now. Okay, yeah. So basically an overview of MRI, it's a tissue response to magnetic fields. MRI detects the RF signals, or radio frequency signals, produced by displaced radio waves in magnetic fields. So the magnetic field in the MRI basically temporarily realigns water molecules in the body. Your body's like 70% water, right? So like applying this magnetic field, it will temporarily realign the water molecules to like a certain direction. And then a second magnetic field in the form of RF waves is going to cause the atoms to produce signals, which are used to create cross-sectional MRI images. So this is kind of like a timeline of how MRI works. So basically the first magnet is turned on, which aligns all the water molecules in your body in one certain direction due to the sensitive proton in that water molecule. And then the second magnetic field is pulsed on and off. And what happens with that is that when like the second field is turned on, the hydrogen atoms like change alignment, and then when it's off, it like relaxes back to the state that it was in based on the first magnet. So like that act of like relaxing back to its original state is different in each like different type of tissue. And when electricity is passed through the gradient coil, it creates a magnetic field causing the atoms to vibrate, and that produces unique signals depending on the tissue that is being like imaged, and that's what's recorded and turned into an MRI image. So this is something that a voxel is like kind of like a term that's used a lot in MRI. It's like a little bit of vocab here, I guess, but it's the smallest unit of volume, and a voxel basically translates into a pixel in terms of an image. So it's a 3D volume in space, basically representing like a pixel of tissue. So a higher resolution of an MRI scanner is going to mean a smaller voxel, and also higher magnetic field strength increases spatial resolution, which previous group talked a little bit about. That's like a big struggle in imaging today, is spatial resolution. But having a higher magnetic field strength and smaller voxels improves the representation of separate structures in the brain. So some of the physics behind MRI. This is a scary looking differential equation. I'm not going to go too much into like how that works, but it's something called the Bloch Equation. So first of all, going back a little bit, when a magnetic field is applied, it hits a voxel of tissue. So like if you think of it as like it's standing straight, and then the magnetic field hits it, the molecule is like pushed to a new position, right? And then it's going to recover back to its natural state over time, and it's going to do that in a spiral manner. And so like the time it takes to do that and the characteristics of this like circle back up is what the MRI is measuring. So how do you know like what frequency of the magnetic field to use? That's where the Bloch equation comes in handy. It basically characterizes the various magnetic fields involved in MRI, and solutions to this lovely differential equation optimizes the excitation of the molecules in the subject for MRI. Okay, so continuing with the Bloch equation, spin states are really important in MRI. This is what, like MRI literally exists because we can capture the magnetic moments due to the quantum mechanical property of nuclear spin. So spins possess this natural frequency that's proportional to that like initial magnetic field. This is called the Larmor frequency. And basically that magnetization of tissue, yeah. Okay, so this is kind of like the next level of understanding MRI. There's like the general thing, okay, there's magnets involved, things happen. This is like more specific. I'm not going to go too much into it, but basically, like I said before, there's this main magnetic field that affects the molecules, and then the second one is pulsed on and off. This is like the rotating part of the MRI, and that is transverse to the initial magnetic field, which means perpendicular, and that's what causes the shift in the protons, and then the relaxation happens, and the last part, decay of magnetization, and the Z component recovery, that's like the tracking it back, like in that like circle coming back up. Okay, so overview of MRI, basically it provides a detailed anatomical view, so not functional, that's fMRI, we'll get into that next. It's non-invasive, if you guys have been in it before, you know, it's like just a tube that you just lay in there. Try really hard not to fall asleep. But yeah, and then some limitations though is that it's really expensive and there's a restrictive design. Obviously, like people who are claustrophobic or maybe can't get to an MRI, things like that. Current uses are mostly diagnostic, so for not just the brain, for any type of like tissue. But like we were saying with the limitations, it's expensive, it's restrictive, it's this big bulky machine. So it'd be really cool if we could get portable MRIs, which is something that OPENWATER is working on. Devin mentioned this company briefly during the first lecture. Basically, they're working on a portable MRI involving red and near infrared light, focused ultrasound and holography, and it's really cool. They can like use light to like look directly into your brain, and it's just like a laser. So we'll see where they go with that. Okay, fMRI is functional MRI. Let's get these two. Basically, the difference with MRI is that this detects changes in oxygen-rich blood that flows to particular areas of the brain. So it's recording metabolic changes like blood oxygenation, and it provides not only an anatomical view, but also a functional view of the brain. So this is just kind of like the components of an fMRI scanner. It looks pretty similar to an MRI. General overview, so obviously this is safe, it's non-invasive, it's an effective method of functional brain imaging, especially compared to other things like x-rays and CT and PET scans that use radiation. This is just like tracking the blood flow basically. Limitations, once again, expensive, a bit restrictive, and it can only look at like the general brain activity, so the blood flow, not at individual neurons. Yeah, current uses are assessing damage, head injuries, or neurodegenerative disorders like Alzheimer's, and also identifying important areas in the brains of people preparing for brain surgery. So doctors don't just like go in blind, they can use an fMRI to kind of figure out like what the plan is, what the map is. Okay, MEG is Magnetoencephalography. Yeah, basically MEG is measuring magnetic fields generated by naturally occurring electric currents. So this is a little bit different. So instead of measuring voltage differences in the brain like EEG does, this is measuring like the magnetic fields generated by those electric currents. So it's kind of like different from EEG. This isn't using action potentials like EEG does. This is using just electrical activity in the neuron dendrites during while they're communicating to each other. So this is measured using super sensitive, they're called magnetometers. And they're usually, these consist of arrays of, they're called SWIDs, which are superconducting quantum interference devices, and these detect magnetic flux. So current magnetometers are really big and they're pretty sensitive to external magnetic fields. The brain's magnetic field is like much smaller than the environment, so it's like hard to have like this big bulky thing. So one of the questions now is, can we use better and smaller magnetometers? And the answer is yes. There's these things called quantum magnetometers. Demario is a researcher. He has a prototype which is like a small cylinder, about a foot long, 6 inches in diameter. It's really small, and it basically contains this synthetic diamond cube that contains like specific special impurities. And those impurities are called nitrogen vacancy centers that bind together and they form this duo inside of the diamond. And these, for some reason, serve as a really good magnetic sensor. And so when a green laser shines on the diamond, those nitrogen vacancy centers emit more or less red light depending on the magnetic field that it's in. And that's how it senses magnetic fields. And so they actually did manage to do this with a squid, ironically, like an animal squid, not the superconducting squid. But yeah, this is really promising in terms of future technology. Okay, PET scans. So PET stands for positron emission tomography. This is basically a metabolic monitoring using biomolecules labeled with radioactive tracers. So biomolecules, the common ones are usually like glucose and like water. Neurotransmitter precursors are also really interesting in terms of like what we're interested in, which is the brain. And then there's tracers involved as well. Unfortunately, they make PET scans really expensive, so that's another story. But yeah, so how PET scans work are biomolecules are first labeled with those radioactive tracers, any specific ones that are useful for the scientist. And those labeled molecules are then injected or inhaled by the patient or the subject. The material travels through into the brain, and then the oxygen and glucose, which are typically the biomolecules that are used, accumulate in areas that are metabolically active. And then the radioactive material breaks down, it emits a neutron and a positron, and then gamma rays are released when one of the positrons hits like a nearby electron, and that like gets detected by gamma ray detectors. And that like reveals the function of the brain. So it's not just like looking like, oh, this part of the brain lit up. It's because of like detecting gamma rays specifically. And when gamma rays are released, when positrons hit electrons, it's called an annihilation event. And this is what releases the gamma rays, generally around 180 degrees from each other. And basically this is detected like, so gamma ray detectors report where these are happening, where these annihilation events are happening, which reveals function of the brain. So overview, advantage, this gives good spatial localization of metabolic changes, and it quantifies like distribution of metabolic changes better than EEG. And then limitations, I'm sure you guys have heard like PET scans are usually not recommended for kids because like repeated use of these tracers can get toxic, and also it's expensive, and then imaging flux with the biological state of the subject. So imaging differs for people like based on whether their eyes are closed or open, or whether they move, or if they're listening to external sounds. So it just like kind of leads to a challenge. It's like what is someone's face like, and then poor anatomical mapping as well. So some current uses, so F18 is fluorodeoxyglucose. It's one of the tracers that is used to image patients with seizures, and this helps identify seizure foci by measuring glucose metabolism. And then it can also diagnose neurodegenerative diseases, for example, Alzheimer's. You can detect like specific plaque accumulation in the brain, and also map out other functions. And then in terms of development, like what's coming for the future, there's improvements all the time for diagnostic information for clinical applications, and then also combining PET with MRI and CT scans. For example, this is mapping a PET scan onto an MRI. MRI obviously has really good anatomical localization, PET scans don't, so putting these together gives you like a really good idea of like what's going on in the brain. And then also a novel application is Ligand imaging with PET scans. So this is basically using, so regular neurotransmitters can't cross the blood-brain barrier, but amino acid precursors to them can. So by tracking these, you can use PET scans to really like understand what's going on with neurotransmitter synthesis and transport and release, and it's just like provides another understanding to like specific neurotransmitters in the brain. So some applications of this, schizophrenia is kind of like a big case. So there was kind of this hypothesis that schizophrenia like symptoms came from increased dopaminergic neurotransmission in the brain, and using PET scans, they were able to confirm this hypothesis. And if you see in this picture, the red area for schizophrenia is much darker than for healthy patients. So it shows that tracking these metabolic biomarkers is actually really helpful in terms of determining causes of diseases like schizophrenia. Okay, CT scans. Basically, the CT stands for computed tomography. It's a 3D X-ray. It's a combination of a series of X-ray images taken from different angles around the body. This creates cross-sectional images of bones, blood vessels, and tissues. And the way this is taken is also a rotating scanner, kind of similar to fMRI and MRI, except it's smaller, I guess. Some advantages: it's non-invasive, it's fast. You have concurrent imaging of bone and soft tissues at the same time. There's high temporal resolution, meaning that it can image your brain in real time. And it also doesn't require the patient to stay very still. And then disadvantages, so there is exposure to ionizing radiation for the patient, and there's fewer details in MRI for soft tissues, and it's anatomical. So some current uses involve examining internal injuries, so blood vessels are really well examined using CT, and also mapping internal organs for treatments, and diagnosing muscular disorders. So one of the examples used for CT, like I said before, was mapping blood vessels. So basically this works by injecting a contrast agent into the patient while the CT machine is scanning. And then this kind of map of a blood vessel or of blood vessels can be stitched onto a 3D model, and then maybe a map of the actual brain, and that once again gives a really good anatomical idea of the brain cells in a specific patient's brain. Okay, last one is optogenetics. This one is relatively new. Basically, optogenetics is using light to control neurons that have been modified to express light-sensitive ion channels. So, so I think I'm going to skip this a little too. But basically, this was started by a couple of researchers, or discovered from Stanford in 2005. Basically, what they did was they found this green algae called channel rhodopsin. No, it was a light-sensitive channel from green algae called channel rhodopsin 2, and they found that this channel, when exposed to light, it would open and let things flow through. So they thought okay, what if we put these ion channels into neurons? And so they did, and when exposed to blue light, those channels opened, positive ions flooded into the neurons, and that makes them fire. So that's super cool. You can basically activate a neuron just by shining light on it. How does optogenetics work? First of all, a genetic construct has to be made containing the opsin gene, whether that's the channel rhodopsin 2 gene or other ones. CHR2 is the most common though. And then a promoter sequence is also added. This is a genetic element that controls its expression. And then this construct is packaged and inserted into a virus, which is then injected into the brain. And then that opsin gene is from that virus is expressed in a subgroup of cells, they can target specific cells with the appropriate machinery to activate that promoter. And then a light is delivered through an optic fiber threading through the skull, and the opsin proteins are triggered by that light at a specific wavelength. Positive ions flood into the neuron, and the neuron is activated. So how can we use optogenetics to image the brain, study the brain, to better understand the function? Here's some studies that have gone on. So there was a rat study where they were able to use optogenetics to manipulate parts of a Parkinson's rat model to figure out which parts actually account for the relief afforded by DBS. So a lot of times with DBS, it's a little bit tricky. They're just kind of sticking electrodes in and making them activate local regions of the brain, but it's not really precise. So using optogenetics, they were kind of able to pinpoint which parts are more which parts are being affected by DBS. And in 2010, they succeeded in a primate. Usually primates are difficult because primate brains are larger, there's like, it's hard to ensure that the virus is getting to all of the neurons and that the activating light is deep enough, but it's really promising that they were able to do it in a primate because maybe it can be used in humans eventually. And then in 2017, something really exciting was that researchers were able to focus like a single, focus laser beams on a single neuron so that like you could target individual cells bearing that light-sensitive molecule, and then that individual neuron was stimulated. So being able to target like a single neuron in your entire brain is also really, really cool. Okay, managed to finish in time. All right, so this is a summary overall of the brain imaging techniques. We went through around 8 of them. Like, just, it's kind of hard to keep track because they all like do slightly different things and it's really nuanced, but like EEG in general, just remember it's like electrical activity compared to MEG, which is magnetic fields in the brain. EMG was like the motor neurons to muscles path, and then MRI, fMRI are both like, so MRI is tissue response to magnetic fields. This is like the anatomical image, and then fMRI is functional. And then PET scans is metabolic activity, CT is like X-ray, and then Optogenetics is activating neurons using light. So if you have any questions, you can come talk to me after. I don't want to keep you guys here for too long, but here's attendance.